Okay, let's do this. So, we are covering how to divide a cell. There are a couple of different things that go on depending what kind of critter you are. If you're a bacterium, it's going to be called binary fission. It is not going to involve a lot of the processes that eukaryotes use because they don't have a membrane-bound nucleus. They don't have linear chromosomes. They have circular chromosomes. And they've got different machinery to do the job. Plants actually divide their cells pretty uniquely to the point where most animal biologists, including myself, don't actually know how they do it. They don't have centrioles, for example, which is part of the machinery to divide an animal cell. But the rest will be largely the same, and we're going to cover the different stages of mitosis. Let's see, what else is up here? We are going to, for the time being, separate cytokinesis from mitosis. Remember, the key difference there, for now, is that mitosis concerns the separation of the contents of the nucleus, and cytokinesis concerns the separation of the rest of the cell. We're going to, for the first time, tackle this concept of chromosomes versus chromatids. Write this down, study it very well. This is going to be the subject of 10 to 15% of the next test, and the success ratio is one of these 25 to 50%. Okay, this is a subject that kills people. Chromosome versus chromatids, and then you throw into that homologous chromosomes. So we have to hit those points hard, remind me. Then I'll talk very briefly about how you should represent your chromosomes with your beads, and I think there's a slide or two at the end that points out what the onion root tip and the whitefish blastula look like. Okay. We have a quantitative experiment in your book that at least one set at your table should take care of, and everybody should understand. So that's the content. Next slide. Here we go. Okay. So prokaryotes, again, no nucleus, no linear chromosomes, no centrioles. All they're going to do is copy everything that's in their genome and attach it to the cell wall and start to stretch the cell apart. This process is called binary fission. I don't know what else you actually need to know for the purposes of this class, except that it is different by those points. Roger, moving on. Okay, so let's see. Oh, and Nima threw in chromatin. What's chromatin, kids? Wrapped DNA. Nicely organized, compacted, getting to the point where you can actually visualize it with just a little bit of stain on your cells. Okay? Chromosome, that is, help me out. I can just keep talking, but come on. What's a chromosome? Kids who've had it. DNA molecules, what? At any stage. I like a good part of that. The key part being it's all one chunk, right? You may know it as an example. How many chromosomes does a human have? 46 is the correct number. We are diploid, so that there are 23 pairs. Yes? Okay. So each one of these, where it is completely separate, not covalently linked to any other pieces of DNA, that's a chromosome. We're going to contrast that with a chromatid. Chromatids exist only in the presence of duplicated DNA. The chromatid, or rather the chromatids, exist after the S phase is complete. And what you end up with is two copies of the same chromosome still covalently linked together. Now here's the tricky part. I'm going to let you finish writing that. The tricky part is you have two chromatids for every one chromosome for the duration of the chromatid's existence. When a when a chromosome containing two sister chromatids, that is replicated DNA, when it splits, your chromatids no longer exist, and you now have two chromosomes where you used to have one. So, to me, the weirdest part about that is even though you have two copies of the same copy of a chromosome linked together, it's called one chromosome. One chromosome, two chromatids. When it splits, two chromosomes, zero chromatids. And this is one of those things you need to memorize at the base, practice it, and beat the heck out of it. For that purpose, I have an exercise online. I'll talk about it later. Okay, so homologous chromosomes exist in haploid or diploid animals. On the count of three. 1, 2, 3. No, let's practice. 1, 2, 3. Diploid only. Diploid is represented as 2N. In humans, in our example, there are 23 pairs of homologous chromosomes. So there are 23 unique chunks of DNA, each of which we have two copies of. One from mom, one from dad. Okay. That becomes incredibly confusing in the process of meiosis when we actually split those up before we split up the chromatids. In the meantime, while we're on mitosis, we're only going to divide everything having been duplicated once. Then this last term, which is just kind of one of those supplemental facts that shows up as multiple choice questions. A conocyte, I would say kenocyte, but I'm kind of perverted by my own research. Or a syncytium is actually, in my opinion, a much better term. Anytime you have a bunch of nuclei in the same single cell, that's what that term applies to. Syncytium is a cell with multiple nuclei. Okay, so not only do you need to know the stages of mitosis, you need to know the cell cycle. And as you practice and study and exercise this stuff, I would encourage you to write down the hierarchy of these things. So the parts of mitosis, prophase, metaphase, anaphase, telophase, those are all under the hierarchy of mitosis. Mitosis is one part of the cell cycle, and its kind of cousins or brothers are G1, S, G2, then mitosis and cytokinesis. Interphase is the whole space of time in the cell cycle where the cell is not actually trying to divide anything. So growth 1, where the contents of the cell are swelling, S, where the DNA is being synthesized, and G2, where we're finalizing the amount of growth that the cell needs in order to split. Those are interphase. Mitotic is anything where the cell's trying to split, including mitosis for the nucleus, cytokinesis for the cytoplasm, and all the organelles and so forth that go with that. Okay, so I'm pretty sure you're going to need to know this for lecture as well. These are the stages of mitosis. Here we have simplified it. We have not divvied up metaphase. What is it? Prometaphase. A lot of times they interject in there. But you need to remember these in order and functionally. I will give a candy bar reward. Is anybody allergic to peanuts? Okay. To whoever comes up with the best mnemonic and presents it to me. If I get one entry, there's one winner. Okay. But anyway, names in order and by function. So prophase is the organization of the chromosomes, the condensation thereof, so you can start visualizing them. Metaphase is lining them up along the center of the cell. Anaphase is where they actually split. And write little stars next to this point. Anaphase is the biggest point of concern for chromosomes versus chromatids. Anaphase is when centromeres that are holding the two sister chromatids together split. There's a whole series of proteins holding those two copies of the chromosome, those two chromatids, which are copies of the same chromosome. There's a whole set of proteins holding those together. At anaphase, they are literally snipped apart, and the cells therefore relieve the tension on those two molecules. Let me rephrase it, sorry. There's actual physical tension pulling on those two copies of the chromosome. And once those binding agents are dissolved or cut, it literally just snaps into position and to the other side of the cell. You'll see that anaphase is actually a really quick part of the cell cycle. So telophase is reforming the nuclear membrane around the new individual copies of the chromosomes, and I still, for my purposes, separate cytokinesis. I do also celebrate cytokinesis. It's a holiday. Into its own part of the cell cycle. Okay? So these four chunks make up mitosis. If you want to add prometaphase in there, that's your own problem. So this is what it looks like in the different systems. In the animal cell, which is our principal concern, but not our only concern, after you have the nuclei separated, I should point this out. You'll notice that in our... Oh, I need my cursor. Oh, I'm sorry. I'll get it now. So, you'll notice that in our cartoon, the nucleus is already formed for this, so telophase is complete, and all we're left with is cytokinesis. We need to divide the contents of our swollen cell. And for that involves the assembly of some microtubules here at the outside of the cell membrane, and you literally just start pulling on a little cytoskeletal string and start cinching. They call it a purse string model. You actually just start cinching the center of the cell, like tightening a belt around it, until the parts of the cell wall get close enough to actually pinch off. Okay? Plants do it somewhat similarly, but through completely different molecular mechanisms, in that they're just going to set up a new boundary and start cinching things off. And the way they do it is they actually form vesicles here at the cell plate. That's a key vocab term there. The cell plate, and nobody wrote it down. The cell plate is the site where these vesicles form, and a new cell wall is going to be born. So essentially what it does is create a bunch of little holes. Okay, so this is actually akin to the exercise that I have for you online. You need to be able to track how many chromosomes, how many chromatids, and how many replicated or unreplicated chromosomes there are at a given part of the cell cycle for, and the matrix continues, for a haploid cell and for a diploid cell with a certain number of chromosomes, N. If you're already an expert, that's fine, but I hope that sounds confusing, and I hope it sounds like something that you need to practice. Okay? But this slide will not let you down, and I always give you the PowerPoints. Shall I go through it, or shall I leave it to you? All right, let's go through it. Okay, so we start with a cell containing 10 chromosomes before mitosis. With these givens, we could be diploid or we could be haploid. Okay? So what this figure does not elucidate for us is homologous chromosomes versus non-homologous chromosomes. If it's haploid, we have 10 individual chunks of DNA with no copies. If it's diploid, we have how many duplicated copies? 1, 2, 3. Again, on the count of 3, if it's diploid, how many chromosomes would you have? What would be the number of N? 1, 2, 3. Murmur, murmur. Okay, so it's a point of unclarity. There will be 5. If it's a diploid animal, there are 5 unique chromosomes, each of which has 2 copies. That's what will be represented by this figure, okay, or by this analysis. So, okay, again, we're not concerning ourselves with homologous versus non-homologous right now. That's for the exercise. All we're saying is that there are 10 before we go through the S phase. G1 to G2. So we've gone through S completely, and there's no ambiguity about whether or not we're mid-synthesis. We now have 10 replicated chromosomes. When I write this out, I like to make this 10 asterisk. because to me, the thing that makes the most sense intuitively is wrong on almost all the questions we'll ask you. The thing that makes sense to me intuitively is how much DNA is there, how many copies of the DNA are there. Okay? And very seldom does the intuitive answer make sense. So I say this is 10 asterisk. It is 10 replicated chromosomes. And what that means is you have 10 molecules, all covalently linked, 2 by 2, each having a copy already attached to it, right? And what that is, and so that gives us 20 chromatids. 10 unique particles, each with a copy on board. Once we start getting ready to divide the cell in prophase, nothing changes. Go through metaphase, everything's lined up. Still nothing changes. The big change comes in anaphase when we have these 20 chromatids split into 2, and they're all in the same cell. They're all in the same nucleus even. They're in a nucleus that doesn't exist, right? So after these are physically split, you now have 20 copies of the DNA. So now it's making sense to me intuitively. 20 unreplicated chromosomes and no chromatids. Chromatids are a thing of the past. As you start to separate these things, within the cell itself, even if telophase is completed, you still have 20 chromosomes in the cell. You just have it compartmentalized into 2 nuclei. I will protect you from tricky wording as to whether or not it's nucleus or cell. But after cytokinesis, we get back to the normal state for the cell, which is to have 10 unreplicated chromosomes, no chromatids. That's exactly where we started. Okay? That cell can go on to go through another phase of mitosis or not. If you have questions, I would love to hear them. Okay. Okay, so this is how we're going to represent things, and we are going to worry about homologous chromosomes today. Homologous chromosomes are present in haploid or diploid cells. On the count of 3, 1, 2, 3. Diploid only. Thank you. And so, how we represent a different copy of a unique piece of DNA is to have something of a different color. You signify that it is a homologous chromosome by its length. So, if this were a simple diploid animal here, I did it on my screen again. If this were a simple diploid animal here, it would have an N of 2. That is, it has 2 unique sets of DNA. There's one. Here's the second. And they have 2 different copies of each. So here's chromosome 1A, chromosome 1B, chromosome 2A, chromosome 2B. Okay? So you'll go through your cell cycle with that. And then these guys down here, same size, same color. Yeah, okay. So when we need to begin our discussion of sister chromatids, we have these magnetic strips that connect the beads. So what you'll actually do is create a second set that goes right here of the same color, and magnetically attach them until you're done with anaphase when they separate. I should warn you on the previous slide that that's the sort of setup you'll see on practical questions. You'll have to interpret, is it diploid, is it under anaphase, haploid, what's going on with the cell. Okay? So then finally we're going to count the number of cells visibly undergoing mitosis, or actually I think over this whole cell cycle, in 2 different systems, in the onion root tip and in the whitefish blastula. These are 2 different systems where the cells should be dividing rapidly. So there'll be 2 parts of that. You'll have to be able to identify what process the cells are undergoing. So like this guy, what would we say that is? Yeah, I think it's somewhere in the metaphase range. I agree. Okay, everybody's lining up at the middle. This guy might be more completely metaphase. Something like this, anaphase, where we're separating. Okay, and when we characterize how many of these cells that we see in each part of the cell cycle. Once we tally those, you can estimate how long each of those parts of the cell cycle lasts, because of our assumption. This is important, because our underlying assumption is that all these cells are actively in the cell cycle. So they're trying to divide, but they're taking time to build up the resources and then conduct the cell division. So what you see is the proportion that are in a given phase represents the proportion of the time that they spend in that phase on average. So we're going to average it over a bunch of cells. And hopefully you can come up with a more succinct way to explain that than I did, but definitely revisit what that assumption is and why it works, because I think that's the only way it'll make sense. And essentially we'll do the same thing okay, and this will just be a practical concern. You'll have to be able to identify the different cell stages again in a microscope or on a slide like this, and also to reinforce the same kind of analysis we did in the previous example. Okay, and then that's the protocol.